Hello and good morning. Today we will be talking about polymerase chain reaction, which is also called PCR. This assignment is presented by Arwa Ashraf, Bishoy Bakhtar, Fadi Nagih, Aslam Ashraf, Marwan Saad, Mina Makin, Mirna Sayed, Muhammad Fuad, Reem Ahmed, and Yusuf Samir. The outline of this presentation consists of introduction, advantages, application, types, principles, components, steps, and references. In 1993, Kary Mullis won a Nobel Prize for what he invented in the 1983. Mullis discovered a way to make numerous copies of DNA fragments in the laboratory, which is the polymerase chain reaction, or which is known as PCR. PCR is a mean to amplify a particular piece of DNA by making numerous copies of a segment of DNA. PCR can make billions of copies of a targeted sequence of DNA in just a few hours. PCR is a mean to amplify a particular piece of DNA by making numerous copies of a segment of DNA. PCR can make billions of copies of a targeted sequence of DNA in just a few hours. Applications of PCR can be used in many fields. It can be used in forensic to solve crimes, in diagnosis of many diseases, in studying evolution of races, as well as creating mutation in studying of fossils across geological time, and for gene cloning and expression. Types of PCR includes methylation-specific PCR, hot start PCR, high-fidelity PCR, in situ PCR, asymmetric PCR, repetitive sequence-based PCR, overlap extension PCR. Real-time PCR, quantitative real-time PCR, reverse transcriptase PCR, multiplex PCR, nested PCR, long-range PCR, single-cell PCR, fast-cycling PCR, assemble PCR, ligation-mediated PCR, methylation-specific PCR, mini-primer PCR, solid-phase PCR, and touchdown PCR. Principle of PCR includes 3 steps: separation of DNA double-stranded template, primer formation, and extension of new DNA strands by DNA polymerase and deoxyribonucleoside triphosphates. The components of PCR includes DNA, that is, the DNA of interest that contain the targeted sequence you wish to copy, a heat-stable DNA polymerase like Taq polymerase, all 4 nucleotide triphosphates, buffers, 2 short single-stranded DNAs that serve as primers, thin-walled tubes, and a thermal cycle, which is a device that can change temperatures dramatically in a very short period of time. PCR process is composed of 35 cycles, and each cycle consumes approximately from 3 to 5 minutes. Each cycle is composed of 3 steps. Number 1, denaturation of double-stranded DNA template. Number 2, annealing of the primers. Number 3, extension of double-stranded DNA molecules. Step 1, which is a denaturation process, is a process that is conducted at a very high temperature at 95 degrees centigrade, where the DNA is denatured, in which that means that 2 strands are finally separated. Step 2, which is annealing or primers binding. It is conducted at temperatures of 50 to 60 degrees centigrade. The primers anneal or bind to their complementary sequence on the single strands of DNA within 1 minute. Primers bind to the complementary sequence of the targeted DNA. Primers are chosen such that one is complementary to the one strand at one end of the targeted sequence, and that the other is complementary to the other strand at the other end of the targeted sequence. Step 3. Primer extension. It is conducted at temperatures of 72 degrees Celsius. DNA polymerase catalyzes the extension of the strand in the 5 to 3 direction, starting at the primers, attaching the appropriate nucleotides, which is A, T, and CG. And finally, the cycles will be repeated, in which the next cycle will begin by denaturing the new DNA strands formed in the previous cycle.